The overall goal of this imaging technique is to monitor fluctuations in luminal SR calcium levels in response to calcium mobilizing agents, specifically in vascular smooth muscle cells.
This method can be helpful in elucidating cellular calcium signaling in a more direct fashion. For example, it's very important to know the relation between calcium transport in the sarcoplasmic reticulum and ER stress. The advantage of this technique is that we can directly measure the calcium inside the lumen of the sarcoplasmic reticulum rather than to have to extrapolate it from indirect measurements of changes in the cytoplasm. Demonstrating this technique will be Gabriela Ziomek, who is a graduate student in my laboratory.
To begin this procedure, prepare a 1 to 25 dilution of protein mixture in DMEM. Next, transfer 200 microliters of protein mixture DMEM to each 35 mm glass-bottomed plate to fully cover the bottom inner glass circle. Leave the plates at room temperature inside the safety cabinet for at least 30 minutes. In the meantime, warm DMEM with 10% NCS to 37 degrees Celsius in a water bath. Afterward, remove the DMEM from the culture flask using a glass pipette. Subsequently, wash the flask bottom with warm sterile PBS to remove the remaining DMEM residue. To dislodge the cultured smooth muscle cells from the flask bottom, wash the flask quickly with